Place your brain with the frontal lobe facing away from you on top of a board that is covered with newspaper and paper towel. Make sure the brainstem is pointing towards you, and if there are any black dots, that'll tell you which end is the frontal lobe. It's actually brain damage from where the sheep have headbutted potentially other sheep. Here we've got the cerebellum and the brainstem. Identify the left hemisphere and the right hemisphere. Identify the cerebellum, which usually sits just underneath the cerebrum. Ignore the split in the brain. The long white part is the brainstem. If we flip the brain over, you've got the corpus callosum, which joins the two hemispheres together. By using the forceps, you'll be able to locate the meninges, which is the membrane that protects the brain. It's a really thin layer on top of the brain. If you're not sure about the texture, I would suggest cupping your hand and feeling the squishy part of your palm. Then you want to find the midsection of your brain and cut down that area and split the hemispheres. Be gentle. Your forceps may be too pointy and sharp and they'll pierce the brain. Be careful when using the scalpel near your fingers. The white area is actually the corpus callosum that joins the left hemisphere to the right hemisphere. It's quite delicate, and you can see just by gently peeling, we're actually splitting the two hemispheres. Here we have the left and the right hemisphere, as well as the glands, the brainstem, and the cerebellum that I'm holding currently. Use your scalpel and cut down the brainstem and your forceps to split it. Are you able to identify the different glands? There's a small little lump, large lumps, lumps at the frontal area, and lumps towards the back. This is the brainstem up close. To the naked eye, it just looks like a little bit of gristle. It's quite soft and quite gooey. Use your scalpel and cut across the section of the brain. Again, be as gentle as you can. Here we're looking at the white matter and grey matter. White matter is the white area and grey matter is the pink area. You'll need to research these to find out what they are. You can see the brain spots up close again. This is the brain damage that we looked at the start of the dissection. Looking at the cerebellum, you need to look at the texture, the colour, the lines. Are they different? How are they different? Why are they different? You can see where it was connected to the brainstem, and it would usually sit just underneath the brain, like so. cut through the cerebellum. It'll be tricky because it's quite small and by this stage quite gooey and soft. Again, it is very hard to see any sort of detail inside. Are you able to identify any other features on one of the hemispheres? The inside of the cerebrum. Now looking at the cross-section of the brain and your cross-section of the brain, try and identify different features. When you're cleaning up, make sure that your gloves go on top of the brain, forceps and scalpel go into the tub, and any other tools that you've used. Then you'll wrap your brain up in the newspaper that you had, like a little parcel. It'll go into a plastic bag, which will then go into a garbage bag, not in the classroom bin. When you've done that, simply wash your board and put it on the rack.